This video is going to focus on the endomembrane system. A lot of people had a little bit of trouble with this system, so let's kind of take a look at it. We're going to work on it nice and slow. This system, once again, is the endomembrane system, and its goal is to produce proteins, enzymes, hormones, any of those that will either be embedded in the cell membrane or will be exported from the cell. So like everything else, it all starts with DNA in the nucleus. And we kind of talked about this the other day. DNA has genes on it, so let's just pretend that this is a gene for a membrane-bound protein, or it's a gene for a protein that will be exported from the cell, or a hormone or enzyme. That gene, after the process of transcription, done with the help of an enzyme, RNA polymerase, is going to make a piece of mRNA. mRNA is created in the nucleus by a process known as transcription. That mRNA is going to leave the nucleus through these nuclear pores on its way to the rough endoplasmic reticulum. So the nuclear pores are going to allow for either ribosomes to get out if they were made in nucleolus, or in the case of this example, the mRNA to leave. Now, in all processes that are a protein synthesis based deal, mRNA is going to leave through the nuclear pores. If it is an enzyme an enzyme that's going to stay inside of the cell, that mRNA is going to go to a free ribosome. The enzyme will be made, or the hormone will be made, or whatever, and it will stay inside of the cell. That is not the case today. Once the mRNA leaves through the nuclear pores, it is going to be on its way to the endoplasmic reticulum, but the rough endoplasmic reticulum. So once again, there is our nuclear pore. Now the rough ER is very, very, very close to the nuclear envelope. And it's the rough ER because it will be studded with ribosomes. So every time you see ribosome, you are thinking ultimately protein synthesis. Once again, a protein can be an enzyme, could be a hormone, could be a protein that's going to stay in the cell membrane. It could be a lot of things. But in this case, we're going to make hormones or enzymes that are going to either be in the cell membrane or leave. So the mRNA is going to leave, and it's going to head to, let's say, that ribosome. Doesn't matter. What's going to be created from that ribosome is a polypeptide chain. Now that polypeptide chain is going to end up in the lumen of the rough ER. Now, unfortunately, I do not have a three-dimensional picture, but the lumen is the space in between the membrane of the rough ER. So it's going to end up in there. It is going to be folded, and it is going to be packaged. So it may get folded, and it's ultimately going to get packaged. And what is it going to get packaged in? Well, everything else, or like everything else, a transport vesicle. So, once again, here is our rough ER, right there, and we have our ribosomes. And what happens is, as this polypeptide chain is formed and it travels through the lumen of the rough ER, it's going to be put into a vesicle. Now, it's called a transport vesicle because that is exactly what it's going to do. So there's the vesicle. And let's make the enzyme, I'll make it purple. Okay, so now you can see this is a one-way street. The polypeptide chain is formed, it's folded, it's put into the vesicle, it leaves the rough ER. This area is the cytoplasm, and that is where organelles are. So rough ER is an organelle, and ultimately we are on our way to the Golgi. Now that's where some modifications are going to happen, more modifications, and we could tag it We could add carbs, we could take away pieces, we could pretty much do what we need to. The Golgi is also going to be a folded membrane, and it's going to also have a lumen. And it's going to have a cis face, and it's going to have a trans face. Now these are important because, once again, this is a one-way street. So in comes the vesicle, the transport vesicle, and in it is the protein, and it's going to head into the cis side. All the while, in the Golgi, it is going to be modified further. Now let's kind of do a zoom in here, so let's make it a little bit bigger. So if that's the vesicle that's leaving, and this is the enzyme or the hormone or whatever, so it's all folded up nice. And let's go ahead and add some kind of tags or some markers. Now the things students have trouble with is this process is pretty much universal for the cell in anything that is going to be put into the cell membrane or be exported. So at this point, the mRNA has the correct instructions, and it's going to send the information of this polypeptide chain through the Golgi. So let's pretend that this particular tagging means that it's going to be exported. Great, that's one possibility. But a different set from different mRNA with a different polypeptide chain may get the tag to be embedded in the cell membrane. So that's the important part. It's now been repackaged. So it came in and it left differently from the way it came in. It is now tagged. It is now ready for its final destination. Now its final destination, or a couple of fates it could have, and let's kind of draw cell membrane as best we can. So we got lipid bilayer there. We'll do a couple of those. Now the lipid bilayer surrounds the entire cell, or the cell membrane. So there's a lot going on there, and there's a lot of this, and there are no spaces there. So again, I apologize on that. So if we have the lipid bilayer, one of the fates of the protein, let's say I could kind of make it into sort of a channel protein, and that protein can be embedded into the cell membrane. It could also be any kind of marker. It could be many things, but that is one possible fate. Another possible fate is to be exported. So if it happens to be, let's say, insulin, or let's say it's a cell from your stomach that is going to release some sort of enzymes, or from your intestines, those have to be exported. So let's draw the vesicle again. So this vesicle is going to come up. Let's make this particular enzyme blue. And the vesicle is going to come up. And it's a more involved process than this, but it's going to fuse. And ultimately what's going to happen is that hormone or whatever, let's happen to say it's insulin, for, let's just say, that is going to be made in a pancreatic cell, and it must be released into the blood. So in this scenario, the two fates are the green protein, which got embedded in the cell membrane, or the blue protein, this insulin, was exported. So those are the two things that could possibly happen to a protein or enzyme or hormone that is made through the endomembrane system. Now, which cells are going to have a lot of rough ER? Well, cells like neurons. Because neurons are going to produce neurotransmitters. Now, neurotransmitters are molecules that are released from a nerve cell, and it's going to release a lot of them. Something from the pancreas. Now the pancreas is also going to be going to release hormones, insulin for example, and maybe some muscle cells. Muscle cells are going to definitely make a lot of proteins that could be used for motor function or for building. So those are the type of cells that would be rich in rough ER. Well, what about smooth ER? Well, we can't forget about that. Now we know about smooth ER as something that is a detox. It's going to detoxify. It's going to kind of help break down poisons, things like alcohol, things like drugs. And you're going to see a lot of smooth ER in your liver. So your liver is really good at detoxifying. You're probably not going to find too much smooth ER in a bone cell because, once those cells are formed, they're pretty solid, and they're going to stay hopefully intact for as long as you don't break them. So if we put the whole story together, I really found this diagram and I liked it. And what's nice is it has the three-dimensional aspect of the rough ER as well as the Golgi. So you can see over here in the nuclear envelope, let's see what color, let's try black. Over here in the nuclear envelope, this is where you're going to have your DNA, which is going to make the mRNA. Now that mRNA is going to scoot right out of the nuclear pores. If you look, the rough ER is right next to the nuclear envelope, and you can see all of these ribosomes. Now that lumen I was telling you about is right here. This is the space called the lumen. And that's where the protein is going to end up going, because what's going to happen is these little pieces here are going to pinch off. This is the vesicle, the transport vesicle. It's going to leave the rough ER. It's going to enter the cis side of the Golgi, where it will be modified. It will then come out the trans side in a different vesicle. And from there, it could go on to be exported from the cell. And you can see here, this is the export. Can't forget that this is the cell membrane, because that's the vesicle fusing and releasing the, in this case, say a hormone. Or it could embed itself into the plasma membrane. So those are the two choices that could happen. Now, another thing that we did not talk about here, but the Golgi could also form vesicles that contain these digestive enzymes known as lysosomes. But we didn't talk about that here. The endomembrane system here, we're going to focus on proteins, hormones, enzymes, and things that are either embedded in the plasma membrane or exported from the cell. So I hope that was helpful, and good luck.